are students that receive travel grants to come here and talk to us. Up next we have Craig Beil. He is on the verge of graduating with his PhD from Colorado State University, and he's here today to talk a little bit about some of his research.
Perfect, thank you. Hello. So my name is Craig Beil. This is just one area of my PhD research. So we've talked about a number of global challenges to wheat production, or production in general. In wheat, we've got new diseases and insects constantly coming in, changing consumer preferences, new temperature and rainfall patterns, and an ever-increasing need to increase yield overall. So with that, we've also got the challenge of limited germplasm base, potentially, particularly within regional programs. And so what my research was interested in is how can we use international germplasm exchange to screen and better adapt to changing climate. So at Colorado State University, we're a land-grant institute, and this is our target population of environments for cultivar release, primarily true hard red winter wheat varieties. And so what we decided to do was look at some of the germplasm that's with Simit and Encarta. And one of the nurseries that jumped out is the facultative and winter wheat observation nursery. So this is coming out of Turkey and representing a number of different breeding programs in Eastern Europe and Western Asia. So what I was interested in is what can we do to better facilitate germplasm exchange between the central and southern Great Plains and this area around Turkey in the Middle East, and then back and forth, how can we do this more effectively? So Simit divided the globe into 12 major wheat production regions known as mega environments. And so the facultative and winter wheat observation nursery is bringing in germplasm from mega environments 7 through 12. And the Colorado environment is mega environment 12, where we autumn plant our wheat, and it needs to be adapted to severe cold. But we've got some of these facultative types in mega environment 7 through 9, which are also autumn planted, but maybe aren't as adapted to as severe cold as it's only moderate cold. So now we've got this problem of low temperature tolerance or winter hardiness between germplasm types. And we see this very clearly in the field. So this is early spring after overwintering in Colorado, and you can see very clearly that there's a genotype effect in these environments, that they're not going to be adapted and they're not going to work for us. So we know that there's two major genetic mechanisms that are going to help adapt wheat to our environment. The first one is vernalization, where winter wheat needs a cold period in order to transition to flower. And then we also know that photoperiod plays an effect, where certain varieties need a long day in order to flower. So true winter wheat types have a long and hardier cold period required. These facultative types or intermediate winter types have a lower requirement for vernalization, and spring types don't have any vernalization requirement, so they'll flower without any cold period. And then we've got photoperiod sensitive varieties that require long days and insensitive varieties. So my objectives were to compare a genomic selection prediction accuracy with including vernalization and photoperiod alleles as fixed effects versus a genomic selection model with only genome-wide SNP markers as random effects. And then second was to identify the vernalization and photoperiod alleles that provide the greatest increase in genomic selection prediction accuracy for winter injury when used as fixed effects in the model. So here's some of the genotypes in the field after, pretty bad winter injury here. And so you can see with that plant population that it's not going to yield very well. So here are the two trial locations that we do in Colorado. Fort Collins, Colorado is our main environment, and that's an irrigated research trial. And then Julesburg, we collaborate with on-farm field trials, and that was a non-irrigated environment. The experimental design, we grew this in 3 years. In 2014, I had 112 of these Fall 1 international genotypes, representing the 20th Fall 1 collection. In 2015, again, I grew the same 112 varieties, but added 88 new genotypes from the next year's nursery. And then in 2016, had 186 fall ones, a handful for a third year of evaluation, some for a second year, and then a greater number for a first year of evaluation in the field. So here's what my trial looked like in Julesburg, Colorado, 2015, and I've got more dead plots than I have a live plot. So this is a pretty serious problem. It's just increased weeding, and then you don't get the same competition between plots when you've got a dead plot. So it kind of messes up your spatial analysis a little bit. So for winter injury phenotypes, it's simply a visual rating of 0% for no injury and 100% for all dead plants. Here's what it looks like by environment. In Fort Collins 2014, there was no winter injury observed. In Julesburg 2014, there was some, but we certainly get a handful of outliers that show much higher winter injury. 2015 was, depending on how you look at it, a good year or not for my research, in that the average winter injury in Fort Collins 2015 is about 48%. And then in Julesburg 2015 is about 85%, and the outliers are actually the ones on the lower end. 2016, again, a little bit more typical, but we did get some winter injury in Fort Collins. Again, kind of these outliers up to 100%. Julesburg, some winter injury among most genotypes, but then we get a handful of outliers. So it works out pretty nice for analysis. Here are the major vernalization and photoperiod genes that we looked at. 2 different winter alleles at VRN-A1, 3 spring alleles and a winter allele at vernalization B1, spring and winter at vernalization D1, and then photoperiod sensitive and insensitive at B1 and D1. So we did genotyping by sequencing on all lines. That gives us about 23,000 markers across all genomes of wheat. But then in addition, we do cast marker analysis for those vernalization and photoperiod loci that I was most interested using fluorescently labeled primers. And then we can identify the individuals that are homozygous dominant, homozygous recessive, and then heterozygous individuals. So what we see just looking at vernalization alleles, the different haplotypes, we have a collection of true winter wheat types, some that are winter wheat with this reduced vernalization allele at VRN-A1, and then a handful of what we would call facultative types in that they have at least 1 spring allele at a vernalization loci. Some of them have 2. So for genomic selection, we do a cross-validation approach where we have a training population made up of 80% of the genotypes for a year, and then a test population of 20%. The training population creates the model using its genotypes and phenotypes. We then put the test population in with the genotypes only to get a genomic estimated breeding value on those individuals. And then for determining how well our model does, we look at the correlation between those genomic estimated breeding values coming out of the model to what their true phenotype measurements were in the field. We run 300 cycles of this, randomly assigning individuals to training and test populations, and then take an average correlation across all of those iterations. So here's what the results look like. So model 1 in the gray box is using only those 6 vernalization and photoperiod loci as fixed effects, while model 2, the orange box, is using 23,000 SNP markers for the model. And so what we see in 2014 is that we get an increase in prediction accuracy using all genome-wide marker effects. Similarly, in both environments in 2015. In 2016, they actually come out even. And then doing a 3-year combined analysis, we get an increase there. So we're seeing that there's more effects in predicting winter injury or low temperature tolerance than just vernalization and photoperiod loci. And then what we did was hold each of these vernalization and photoperiod loci as a fixed effect by itself. And what we see is that we get an increase again when we hold VRN-A1 as a fixed effect in 2014, a little bit in 2015, a decrease in 2016, but then a little bit of an increase looking at a 3-year average across environments. VRN-D1 was also shown to be pretty effective when treated as a fixed effect. We see an increase above the genomic selection prediction model here using the 23,000 markers, again in 2015, and in 2016 actually a pretty nice increase above that model and across the 3 years. And then we see that photoperiod loci really don't have an effect for us. It actually decreases below model 2 accuracy in all of our situations. And then we also looked at using these as multiple alleles as fixed effects. So for VRN in the blue box, it's actually holding all three of those vernalization loci as fixed effects in the model. And so what we see is we get an even bigger increase than we would with any single allele as a fixed effect. And that, I guess what I should also point out is that these prediction accuracies are pretty high. When genomic selection is done in yield, sometimes it's 0.2, 0.3. So we're up around 0.7 and close to 0.8 in 2015. So again, in 2016, where Model 1 and Model 2 came out the same, including these VRN alleles as fixed effects, actually helps the model quite a bit. And then again, with holding vernalization and photoperiod alleles, or just photoperiod alleles as fixed effects, we don't see too much of an increase in prediction accuracy, or it's not consistent across environments. So the main conclusions are that the fall 1 genotypes used in this study represent both facultative and true winter wheat types. And they showed this not only in their low temperature sensitivity, but also in their allelic combinations at vernalization and photoperiod loci. Genomic selection accuracies were high enough for us to allow for prediction and selection of fall 1 genotypes in future years, including the alleles at the vernalization loci as fixed effects increased our genomic selection prediction accuracy over a GS model where we only had all marker effects as random. And including photoperiod loci as fixed effects really didn't do anything for our genomic selection prediction accuracy. So our applications for this in the breeding program at Colorado State is hopefully we can decrease the loss of lines in field trials because there's additional inputs needed with that. Genotyping lines is becoming cheaper than phenotyping, so hopefully we can just genotype these fall 1 nurseries instead and pick the best 10% instead of having to grow them all out in the field and increase costs. And then also, what's nice with genomic selection is that the unpredictability of winter injury screening in our environments isn't always there. So now we can use this historical data as a model and predict going forward. So with that, I'd like to thank my advisors, CIMMYT for their collaboration, and the Colorado Wheat Research Foundation. Thank you.
Any questions for Craig?
Yeah.
Were the SNPs that you used within the genes previously validated causative SNPs, or are they linked to they're just within the gene?
Yeah, just randomly within the genome of wheat. So that's what's nice about genotyping by sequencing is that you just get a nice random sampling.
So what I say about the cat forms, the ones within the vernalization?
Yeah, those ones are known. Those ones come from the literature, and they're usually a causative SNP or an insertion deletion.
Do you have any GBS SNP nearby gene?
Yeah, I'm sure we did. I didn't look at it specifically, but the genome's pretty saturated, so it's good. And then I think what it's showing is that, again, with the GBS markers, it's picking up all the genetic effects, or most of the genetic effects that might control winter injury or low temperature tolerance in addition. And then we're placing specific emphasis on the ones that we know are known vernalization alleles. So there's a little bit of overlap, but yeah.
Any more questions?
All right, thank you.